Log on, tune in, find out. Another good idea from Cambridge.
Good morning, everybody. It's my pleasure to introduce Arne Mooers as the first speaker of today. And Arne, it's your stage.
So, is that working in the back? Yeah? So I changed my title just a bit because now I realize I'm talking about private, not public information. A private good. So these are trees as we normally think of them. This is work. So I offer the biological sort of sugar coating over all the heavy lifting which was done by all the other people you see in this list. Well, some of my former students, and of course Mike's been focal to a lot of this work. Now, the motivation for this, there are two motivations. One is this, and this is, as a conservation biologist, which I've become, I guess, we're in a situation where not all species can be saved from extinction. We talk about prioritization and the word triage. So the idea being that we're not going to be able to conserve, preserve everything, so we have to manage biodiversity in some sort of smart way. That's the first motivation. The second motivation is my background, which is I'm still sort of fascinated by the shapes of actual phylogenetic trees. So what do they actually look like, and what can they tell us? And an insight. This phrase about what a tree is actually comes from Wade Madison when I was trying to explain what I was doing, and the idea being that the tree, not only is it a representation of who's related to whom, but it actually represents, this is why I use the word information, shared and unique information concerning solved evolutionary problems, right? So natural selection and other processes, particularly we're interested in traits that fit the environment from a conservation perspective. And so you can think of the tree, in this case a rooted tree, as a graph representing evolutionary information. So it's not, minus p log p information, it's evolutionary information. But we'll get back to that. So those are the two sort of inputs. We have to come up with a smart way, or we should. It behooves us to come up with a smart way to prioritize bits of biodiversity for management, and we actually have a representation of information, evolutionary information. So in the next 45, 50 minutes, I don't know, I've got 94 slides, we'll see. If we believe that multicellular life can be represented by a tree, that tree contains something, gives us some information about evolutionary information, what do those trees look like? And that is relevant, you'll see why. I hope there's a couple of parentheses in this talk. I hope you'll see why it's relevant. And why it matters what they look like. And given that they have a certain shapes in general, it might behoove us to think a little bit more about them and how we can use them for conservation. So I'll give you some pictures actually of animals and things, and some sort of practical examples of how we can use trees for conservation. Okay, so the first parenthesis, and that is, what shapes might phylogenetic trees actually have? So from theory, back in 1967, and maybe a little bit before, but there's a number of papers that came out right around this time, A.W.F. Edwards here at Cambridge with Cavalli Sforza presented the Yule model as a prior. So the Yule model goes back to 1924, and we've heard it mentioned a number of times already this week. And it's actually presented as a prior on tree shape, i.e. being it might be a good null model when you don't have any data as to what trees actually look like. 1996, this was generalized to the homogeneous birth-death model, which we've also heard in Alexi's and Sarah's talk yesterday, by Zhang and Ranala. Now, when the Yule model was proposed in 1967 as a prior for tree inference, it was actually too hard, so it was never used, surprisingly. The homogeneous birth-death model, which is sort of a Yule model is a special case of that, is actually now a prior that's actually used in things like beast, like we heard about yesterday, i.e. people think of it as a reasonable representation of what phylogenetic trees actually look like. Okay, so we do know a lot about Yule trees. As I say, it's been around for a long time. If you have it has a single parameter, which is the rate at which new lineages give birth to other lineages. So you start with 1 or 2, depending on your perspective. You go along, and there's some rate at which you split, and then some rate at which you split. And it's the same. It's homogeneous all through time. It's homogeneous across all the lineages. It's an exponential process. So we know, for instance, that after a certain amount of time, we have an expectation for the number of lineages that will be around. If we actually know what this, oops, now I can see now why people are doing this all the time. If you actually know, if you don't know what the birth rate of lineages is, you can actually estimate it if you know the tree length, and simply n over 2 over the tree length, and we've known this for quite a long time. And if you don't have the tree, but you actually all you have is t and n, you can also there is actually a maximum likelihood estimate of the birth rate, right? So if someone says, I have a genus, it's 20 million years old, it has 64 species, you can calculate maximum likelihood estimate of lambda without knowing anything else about the tree. Very useful properties. We don't know everything, though, or at least we're not aware of everything about the Yule model. So if lambda is the speciation rate, right, and it's a sort of a Poisson process, and the expected age of any lineage will be just 1 over that speciation rate. That we know. So we might naively think, and I thought, and everyone else thought, that, or at least no one said anything to the contrary, that the average length of a branch or length of an edge on a tree, on a Yule tree, that was produced with rate lambda would be just 1 over lambda. That would stand to reason. It's not actually true. So there's an elegant proof by Olivier Gasquel that the average length of an edge on a Yule tree is actually half of that. So the average length, if you just take an edge from a tree, you don't know, just randomly, you don't know where it comes from, on a random Yule tree, its length is actually 1/2 of the expected time between splitting events. His proof draws on the memoryless nature of the exponential process, the idea that if you show up at a bus stop and they come randomly every 20 minutes, and you say, well, when did the bus last come? If you show up randomly, you might naively expect that they came only 10 minutes ago, but actually came 20 minutes ago. We did actually know this already, if we'd actually looked at the equation, because the tree length is actually the average length times the number of branches, so the number of branches 2n minus 2 on a rooted tree, and if we have the average length, we get the tree length. Well, we can rearrange that so that we actually now estimate this, which is n over 2 over the tree length. Tree length is this 2n minus 2 minus the average length, and so we simply rearrange for average length, and we already get 1 over 2 lambda. So it was already there, but no one had actually noticed, nor had they offered a formal proof. Okay, so that's kind of interesting, but we'll just park that for a while. That's kind of interesting. What about the homogeneous birth-death model? It has 2 parameters. So it has a birth rate, which is lambda, and it has a death rate, which we call mu. So yesterday, from the beast talks, lambda they actually called beta, and mu they actually called gamma, which is kind of unfortunate because I'm also going to talk about gamma. But those are the birth and death rates, lambda and mu. And there are many, many complex expressions for the expected some of the properties of those trees. And for instance, luckily, there's a very simple one again for the expected number of lineages after time t, and it's just the net rate of diversification, so lambda minus mu multiplied by t. And Hang Lang on his poster has a nice derivation of this. This is a well-known formula, but he's derived it in a new way out here in the poster. You might take a look. And Sean Nee and Tanya Sattler and others have analyzed this model quite a bit. Now, the 2 models have the same expected distribution on topology, so the same shapes. So topology, so not the edge length, just the shapes. They're the same, which is useful. They do not have the same distribution of the edge lengths, especially as you move from the past to the present. Now, there's many ways of representing edge length, distributions of edge lengths on the tree. A very simple one is using this gamma statistic, which is just this, which looks complicated but isn't really, from Pybus and Harvey. And what you can do is you can get a simple number that represents sort of the waiting, actually represents the waiting times from the root to each next splitting event. So you go from the root to the first splitting event, to the next splitting event, could be anywhere in the tree, to the next splitting event, all the way up until the present. The interesting thing about birth-death trees is that if you actually plot sort of the splitting events, so you go from 2 to 3 to 4 to 5 along here, as you go from the root out to the present, this is the sort of shape that you get. So you increase, and then there's this upward trajectory. And that upward trajectory, which means that the waiting times as you get nearer the present get shorter and shorter and shorter, leads to a gamma greater than 0. The Ewell tree, because this was created, this gamma statistic was created to test against the Ewell tree. The Ewell tree is actually a perfectly straight line in semi-log space, and gamma is in fact 0. Okay, so you can just take a tree and you can calculate gamma on it, and you can ask, well, is it, does it look, is it around 0? Is it much greater than 0? Or is it much less than 0? And the reason I tell you this, when I said what do trees actually look like, and I didn't, so this is, I've wanted to do this for many, many years, but I didn't believe the trees and how, especially the edge lengths on trees, and I said, well, we really have no idea what the waiting times on trees are. Other people basically just went out and did it. And if you actually go and you collect large samples of trees, so 200, 300 trees that are reasonably well sampled, Mark McPeak did it first, and then Morland did it just recently in PLOS biology, and you fit the models. Well, the first thing, you can fit a bunch of models to them, but the most easiest observation is that that's what they look like. Gamma is actually less than 0. So the waiting times for splits on the tree get longer and longer as you get near the present. So another way of thinking about it, though it's not strictly the same, is that the pendant edges are longer than you might expect under a birth-death model. And even longer than you expect under the Ewell model, where there's no extinction. Now, of course, there's lots of extinction because there are lots of fossils, so I'm not suggesting that the Ewell is telling us how evolution actually happened, but I'm presenting it as an abstraction of what trees might look like. Okay. So inferred trees have too long pendant edges, or the waiting times are too long as you get near the present, relative to homogeneous birth-death models. Why might that be? Well, there are a number of reasons, and some of them probably instantly pop into your head. So if your model is misspecified, so these are inferred trees from molecular data with relaxed molecular clocks or other sorts of massages to produce ultrametric trees. If the model is misspecified, as you know, even from parsimony, pendant edges tend to get longer, right? So homoplasia gets pushed to the front. And that happens under likelihood as well. So Dan Roboski was the first to point that out. Lars, in his talk day before yesterday, made the same point when he was looking at compatibility and how the interior edges get longer as you get better data. Non-random sampling of leaves can also give you this phenomenon. So if you, and I don't know to the extent that this is true. So this is just a potential problem. But of course, if you don't have all the leaves in your or all the species in your sample, but you make sure that you capture the root and the splits that are near the root, right? Then you're going to get a lot of the structure near the root, but you're going to be missing a lot of the splits near the tips, which will make the pendant edges look longer. Or, and most intriguingly, negative gamma may actually be the truth. It's intriguing because if negative gamma is the truth, then the samples of trees that we collect and publish are obviously a non-random sample of the tree of life, right? Because you're seeing a slowdown, so the node that you start on is actually slowing down, and it can't be slowing down. It's not fractal in nature, so that means that we're looking at a non-random sample, and furthermore, the homogeneous birth-death model, which is now the standard prior for tree inference, is incomplete. There's something missing. So to the extent that gamma actually is negative, we have to think a little bit more about what our priors should be just for building trees and also for estimating them. I think there's going to be a talk in a bit about how you estimate lambda and mu on trees. Now, interestingly, not only do we know a little bit about the waiting times, well, to the extent that gamma is negative, we know a lot more about the actual topologies, the actual shapes. And this is old work, but it's still it's going to become relevant later again. And if you actually go out and collect thousands of trees and you look at them, they tend to look more like this than like this. And they look more like this even relative to the Ewell expectation. Right? So they're even less balanced than you would expect under a homogeneous process. Now, why is that relevant? It's relevant for conservation and I'm sure you'll make that connection later. But it's also relevant because models that give you negative gamma, so process models that give you negative gamma, it's not homogeneous birth-death processes, but other processes that, so there are models that do allow for negative gamma, don't give unbalanced trees. And models that give you unbalanced trees give you very positive gammas. So this is sort of a challenge to people who like thinking about this kind of problem, is that we do not yet have a general process model that produces both unbalanced trees and that produces reasonable edge lengths, negative gammas. But people are trying. So I think the most recent or most interesting sort of approach to this is actually using neutral theory of biodiversity. Now, neutral theory of biodiversity in its present incarnation, if you know anything about it, produces ridiculously high gammas. So absolutely has nothing to do with reality. But there may be reasonable biological modifications that may produce more believable edge lengths. The neutral theory of biodiversity does produce trees that seem to show the right property in terms of the variation in speciation and extinction rates across the tips of a phylogeny. Okay, that was a parenthesis. So this is where we're starting. How do we move from there to trees? Edward O. Wilson, very influential conservation biologist, taxonomist, systematist, suggested that the aim, I should say one aim obviously, but that he actually wrote, the aim of conservation biology should be the preservation/management of the information content, that's his phrase, contained in the DNA of all species on Earth. Okay. Now I don't know how precise his definition of information is there. But if we believe that the tree represents shared and unique information about evolutionary problems, then we could make a logical leap and say that the tree may also represent Wilson's information content. All right? So now we actually have a definition of what that tree is. It is shared and derived, shared and unique information content, which means we should attend to the tree, right? So if we just add up all the branch lengths on this tree and we pretend those branches represent information, then Wilson's recipe then is to save as much of the tree as possible, and if we just add them all up, we get total information. That, according to Faith, we can call phylogenetic diversity. So PD then refers to the total information of a subset or a set of species that are, because they're connected or as they are connected via a rooted or unrooted tree. Okay. So that must be our goal. Then our goal then, as a conservation biologist, is to attend to the tree when you're choosing which species to, in a triage situation, for instance, which areas to set aside for parks or which species you should actually invest in with our money to save them. Okay, there's a problem, or there's a perceived problem, and that refers back to this idea of what the trees actually look like. My mentor, Sean Nee, so he's on my committee, wrote a very influential paper with Bob May back in 1997 that's been cited over 150 times. Remember, now this paper is a modeling paper, it's not a techniques paper, and there's no data. So actually 150 citations is pretty good, right? Because there's no new technique in here. And the claim that was picked up is that you can lose 95% of all species on a tree and retain 80% of the tree, i.e. retain 80% of the total information on that tree. That's the quote from the paper. That's been picked up, so picked up by Sean. In a soundbite, 80% of the evolutionary history of a clade can remain after the extinction of 95%. Paleontologist Doug Irwin, preceding National Academy of Sciences, the most direct demonstration of redundancy was a study that showed, etc. Andy Purvis, also in annual reviews, phylogeny is surprisingly robust to random species loss. Janova Milsey in Ecology Letters now, and Santos in PLOS One, goes on and on. So this is percolating through the literature that you can lose species at least randomly from a tree and retain a lot of information. This is the tree that was modeled to make that statement. This is the most extreme homogeneous birth-death tree imaginable, because this is actually a coalescent tree where lambda and mu are equal, right? Now, in order to get this tree, of course, you have to actually condition on n, which is what happens when you have a coalescent, because you're actually conditioning on a sample going back. You're not actually growing a tree up. You cannot grow a tree with lambda equal to mu. I mean, you can try, but you need lots and lots and lots and lots and lots and lots, and lots of iterations, because almost all of those trees will, in fact, go extinct before you reach any reasonable n. People didn't, the connection between that statement and this tree isn't apparently obvious in the paper itself. So, if you actually calculate gamma on a coalescent, it turns out, and Mike proved this a couple of weeks ago, that it's actually arbitrarily large. It actually increases with n. So as n gets bigger, the gamma, so you can think of it as the relative proportion of the pendant edges to the interior edges, it becomes arbitrarily large. That's just the equation for the expected gamma on a coalescent tree of size n. It increases with n. Okay, what about on a Yule tree? What's the so think of this then again. I should have made this more precise, but you can think of this again as the relative pendant edge length to interior edge length. So if you look at the two and you say, well, are they different, or is one on average larger or smaller than the other? What about on a Yule tree? So we know that the average length on a Yule tree now is 1 over 2 lambda, but are pendant edges longer or shorter than interior edges? Well, let's intuit a little bit. So if you so the interesting thing, or sort of the insight that Mike and Olivier have made, is that when we show up, and Tanya as well, when we show up to sample a tree, like in the present, we're actually truncating that tree, that ongoing evolutionary process at the present when we show up. So that has the effect of sort of truncating the length of those pendant edges, right? They haven't lived out their life until they either go extinct or speciate again, because we show up at some arbitrary time. But the insight is that it also truncates the interior edges, because the interior edges, if they were too long, they would have become pendant edges. So we're actually sampling shorter than the universe of interior edges by showing up at some particular time. Because those interior edges that would have be long, so in the tail of that exponential distribution of waiting times or lengths, for instance, would become pendant edges. So the only interior edges that we see are the ones that actually split earlier than you might expect. That may be wrong, but that's the way as a biologist might think about it. And it turns out that the expected length of a pendant edge is almost exactly the expected length of an interior edge on a Yule tree. So very different from a coalescent tree, almost as different as you can get. The exact difference depends on whether you condition on n when you show up, or you condition on the age of the tree, but they converge very quickly. So Tanya has shown that if you condition on n, and n is not too small, then it's exactly 1 over 2 lambda, both the expected pendant edge and the expected length of a pendant edge and the expected length of an interior edge. If you condition on t, it becomes a little bit more complex, but they do quickly become the same, because these terms become quite small very quickly. And if you take the expected value, so if you condition on t, and then you use the exponent of 2 exponent lambda t, you get exactly 1 over 2 lambda as your expected length for both pendant and interior edges. Okay, why am I telling you all this? Well, you guys are mathematicians, it's kind of cool, but it's actually relevant. If you just do simulations, you see that the sum of the total pendant edges, because this is the easiest way to do it, over the sum of the total tree, of course if you only have 2 edges, they're both pendant, so there is no interior edge, but very, very quickly, even at 200 tips, you very quickly get this sort of convergence that pendant edges and interior edges are the same length. Okay. Hae-Young Lee, in his undergraduate thesis, was able to show that even if you have moderate extinction, so this is just lambda over mu now, so this is where extinction is very high, so less, even with moderate extinction, so these are now homogeneous birth-death trees, which I'm telling you I don't believe in, but even if you allow for them, pendant edges and interior edges are pretty much equal. So here, for instance, you get they're about 80%. The pendant edge is about 80% of an interior edge, with sort of lambda mu of the ratio of 3 to 1. So even if you believe, if you want to believe homogeneous birth-death processes, you still don't have this extreme difference between pendant and interior edges. Okay, so let's go back to extinction. Nee, in that influential paper, gave an expression for the amount of the tree remaining. So PD, remember, is the sum of all the edges, the amount of PD remaining, if you randomly select k of n species to remain in the tree, so to survive. As you can see, it's actually in terms of k and n separately. Mike was able to do the same for a Yule tree, and it's this expression. But here, it's always in just the survival probability. So it's not actually in n itself. It's always in k over n. So we can actually write this as just the amount of tree that's remaining. So mu again here, sorry. But the PD remaining, then it's just minus survival probability log s over 1 minus s, which means independent of tree size, right? N doesn't show up in there. So, but for the coalescent, it does. So if we actually, so let's take a tree of 10,000 tips, 10,000 tips, and you change the survival probability s, and you say, well, what's the expected amount of tree that remains, right? So this is the sort of you're saying as you have random extinction, how much of the tree remains. And as you can see, you can have a very low survival probability, and you retain a lot of the tree. So 80% of the tree, even if only sort of 20% of the species remain on this tree. Now this, because of this sort of the way that the pendant edges become sort of arbitrarily small as you increase n, can actually get steeper and steeper and steeper as you simply increase the size of the tree that you're working on. On a Yule tree, it's completely different. It's almost linear. It's not linear. There is redundancy, of course, but because pendant edges are the same length as the expected interior edges, the first ones that come off take off, 1 over 2n minus 2 on the entire tree. And so this is the loss of PD under the Yule model. It's very different. Now, again, Hae-Young and Mike were able to extend this expected loss under random extinction to birth-death trees. Here's the expression. Here, where lambda, as I remember, I said that even with moderate amounts of extinction, the trees are pretty much the same size, same shape, and you can see that the extra loss that you get is very minimal. Even if it's 90%, so even if extinction rate is 90% of the speciation rate, there's still sort of a fairly steep loss in total information. Remember, loss of the tree as species go extinct. So there's something really interesting going on here, that the first 90% in terms of the relative amount, or the first sort of 9 to 1 extinction to speciation rate here has very little effect, but the last bit has a huge effect. So it's very nonlinear. So this would argue that the homogeneous birth-death process is okay, but so I don't want to contradict myself without making it clear that I know I'm doing that. But the point being that it's very different from this statement. So and the statement was made on a clade of 5 million tips, but they could have had 100 million tips, and then it would have been, choosing 0.1% of the species to retain would retain 99% of the information, right? It's an arbitrary statement. And of course, under the Yule tree, where gamma is equal to 0, it's independent of n, and if you lose 95% of the species, you actually lose over 80% of the total information, so much closer to linear. Right. So all that means that all the stuff I've done before and that I do actually might be relevant, because if it was 95%, then who cares which species you retain, if your goal is Wilson's goal of retaining information content. Okay. So Yule should be so wave my hands. Yule should be conservative to the extent that gamma actually is negative, not 0, right? And this I think is interesting. I have no idea how to tackle this, but it's it's possible and probably probable that the information content, however we measure it, degrades over time as it's overwritten by evolution, right? New information, new mutations will override old information. Adaptation is constantly going, which means that the interior edges, if they're supposed to represent information, should be compressed further, right? We should be weighting exterior edges more than interior edges if we're interested in information. But how you would actually test that, I don't know. I'm hoping that we can do it with entire genomes, but larger intellects than mine will have to take that on. So we forge on. So forget those guys. Now we're going to. I'm now going to give you a couple of examples where this kind of information might be used. One is where you actually map evolutionary information, like literally map it on a map, and the other is where you map it onto species, which is the well, I shouldn't say it's the more important, but it's the one that we've taken farther. Okay, so I'm going to give you an example. So here is an example. Lemurs of Madagascar, great group, lots of species at risk, self-contained, know lots of information about them. They're cute. This is Madagascar. The green is the forest where they live. We can build a tree. So we actually got genetic data for 57 of the 67 recognized species. On that group, before we add the other species in, the edge lengths, the pendant edge lengths, are almost the same on average as the interior edge lengths, so it looks very much like a Yule tree, not like a homogeneous birth-death tree, even though there's been lots of extinction, we know that in lemurs. And we were able to use some of the tricks about Yule trees to actually place the other 10 species. So we have a complete tree, right? So now we have all the genetic information, or evolutionary information, represented for all lemurs on Madagascar. What can you do? Well, you also know where they live. So here's just an example. Let's say we've got 3 species that are connected in this way. A lives in these 2 quadrats of the island, B lives in all 3 of here, and C lives here. Well, what you can do is you can cut the tree up. So you take the same total amount of information, simply cut it up by the number of quadrats in which an edge is found, and then you sprinkle those partial edges onto the tree, onto the map. So now you have the entire tree chopped up and laid down onto the map. That's what we've done here. So this is, B is found in 3, so it's found in the 3 quadrats where it is found. Its information is then distributed amongst those 3. And then you can say, well, which quadrat contains the most information, right? That's the focus. That's where most lemur information, or that's where most information in this tree is actually found in Madagascar. So you get these types of maps. So the red now is where the II is found. The II is found here in all these quadrats. This Sophaco is found here, and this one is found only up here, right? So its entire information, so the entire pendant edge of that species is found exclusively in that quadrant. So you can produce maps like this. So the red represents where more of the tree is found, and the yellow represents where less of the tree is found. This is just species richness. Doesn't show much discrimination, because many go into a bit of Madagascar. I've never been, but you go into a bit of Madagascar along the coast here, you're going to see between 8 and 10 species, different species, but on average between 8 and 10 species along here, for instance. But if you actually look and see, well, if you include who they're related to and where else they're found, you actually get a lot more discrimination amongst the places, right? Because you're including more information. We can take this one step further. Is we can also project the information into the future, at least in theory. So if someone comes along and says, well, this species is critically endangered, and there are lots of them in Madagascar, and in the next 100 years, there's a 9 out of 10 chance, 90% probability that species would go extinct if we don't do anything. Right? We can project its trajectory. For species B, we can say, well, it's got 6% vulnerable. It's only got a 1 in 10 chance of going extinct in the next 100 years. And this actually is what people say. These are kind of extrapolated, but this actually is the definition of vulnerable at the IUCN level, which is the group in Switzerland that actually comes up with these numbers. And so you can actually say, well, given that, we can actually now calculate the survival probability, little s, right, or the survival probability of a particular edge, which is 1 minus the probability of extinction. And of course, the survival probability of this then is 1 minus the extinction probabilities of those 2. So they both have to go extinct above for the interior branch to go extinct as well, to be lost as well from the tree. So we can actually just translate the actual tree with the probabilities of extinction to produce a new tree, which is weighted, and we can call that the expected PD. So that's the expected amount of information projected into the future. This has a long history in different contexts, Witting and Loschke particularly, and then Mike Steele and a bunch of his students have sort of extended this further. This is quite interesting because now we can actually map that tree onto the landscape. We could. What we can also do, though, is we can produce another mapping, another weighting, which we could call imperiled PD. Okay? And that is just 1 minus the survival probability multiplied by the edge lengths. Okay? So instead of taking the survival probability, you actually take the extinction probability. Now, what is that? Why would you do that? Well, the first thing, it's obvious, that this mapping is just the total minus the expected, right? These two must add up to this, because it's just 1 minus S versus S. And this actually is the information that we expect to lose, not the information that's going to be around, but this is the information that we expect to lose, but it's actually distributed on the tree. It's not the total information, it's actually distributed among the edge lengths, right? So we can produce that tree. This is what it looks like. You'll see that there's the interior edges are pretty safe, right? Because you have to have all the species above go extinct before you lose an interior edge. The interior edges become pretty safe. We don't expect to lose the interior edges, but there are lots of, like for instance here, there's an interior edge that's subtended by 2 species that are both at risk of going extinct, and therefore we expect to lose a lot of that amount of information. You actually map that onto the landscape. And so if you map that onto the landscape, you do exactly the same thing, you might get a different answer as to if you just looked at PD. You're now looking at risk PD onto the landscape. And you get even more discrimination. So here's the phylogenetic endemism, right, which highlighted, and here you highlight others. So this is not just the species, that's the information, this is the information that is currently at risk. So I'm not suggesting, we don't suggest, this is in review, we don't suggest that we should actually use this map for conservation yet, but the idea is that you should eventually use maps like this for thinking about managing areas in which species that you care about live. It's called imperiled phylogenetic endemism, at-risk information on the landscape. Okay, well, the 2 issues with this exercise, it produces a map, produces a layer, but it actually doesn't have anything to do with complementarity. So you can't say, well, if I preserve the first one and the second one and the third one, I'll get the maximum amount of total PD preserved, right? So it doesn't trade off in a complementary form. But more importantly, it actually deals with PD, and there's no conservation organization or piece of legislation that actually cares about groups of species. They actually care about individual species. So individual species work when you're managing things, like in zoos or on farms, and they actually work for law. So for instance, the US Endangered Species Act, which is the most influential one, is very clear and explicit that taxa that are most genetically distinct should be given priority. Most genetically distinct taxes, I'm reading just the red, is of greater significance than the loss of a most genetically distinct taxa is of greater significance than the loss of the least genetically distinct taxa, i.e. we care in law about how many close relatives you have. That's unique genetic evolutionary information. So to the extent that species and not subclades are the focus of conservation, we have to map that tree back onto the tips. Okay? So we have to ask how species complement each other on the tree. There's lots of different ways. You can complement the entire tree, that's just the pendant edge. You can complement a set of species you've already defined, and that would be the phylogenetic diversity of the set. You can complement all leaves in the pairwise fashion. I'm not going to talk about that, but if you're interested, there's lots of interesting math associated with this particular measure. And then the 2 that I am going to talk about is that we can actually ask, well, how does a leaf complement equiprobable subsets of leaves? Right? How much information does it bring to the table, to some random table of the other leaves? Or how much information does it bring to this expected future subset of leaves, this expected PD that I introduced when I was talking about the lemurs? Pendant edge, you know what that is? That's just this. Now, so what we're saying is that this species brings this much new evolutionary information to this collection. Right? That's what I mean by complement. But if the only other species that's out there is this one, then this is how much information it brings. If the only other species out there is this one, then it actually brings a lot of information to the table, at least in the rooted case. And you can actually go through all the possible subsets that a particular species could complement, and you get something called the Shapley value. And the Shapley value then is the average amount of information that you bring to an average random subset of other species. And this is a Shapley values from 1953, but this was applied to trees published in 2008. Now, the funny thing is, I see Dave Redding's here at the back. Dave Redding, when he was doing his master's thesis in Norwich, I believe, actually came up with a completely different ad hoc measure of how you apportion a tree to the leaves. And he said, well, why don't we just say x gets all of this, and x and y actually share this branch, so I'm going to say half of the information I'm going to give to x, and half of the information I'm going to give to y. And there are 4 species that sort of share this amount of evolutionary information, so I'm going to give 1/4 to this, 1/4 to this, 1/4 to here, and 1/4 to this. We call that sort of the fair proportioning of the information to the leaves. And it can be represented, obviously, trivially. Well, the weird thing is, and Akimamoto, who is an undergraduate in my lab, who was working with Dave, noticed this, is that the Shapley value and the ED are almost exactly the same. Well, almost exactly the same. On this particular tree, they are the same. We asked Klaus Hartmann, who's doing his PhD with Mike, what's going on, and he first showed that in general, if you produce a bunch of Yule trees, for instance, and do this, you get a very, very strong correlation, so R squared is greater than 0.99 all the time. He also proved that as n becomes large, they converge exactly. The weird thing, of course, is that Dave and I, and conservation biologists, think in terms of rooted trees, but the Shapley value is unrooted. Right? It's a very interesting sort of confluence. Now, the ED of Shapley has some other interesting properties. If you actually choose species, so you take your species, you take a tree, and then you calculate the value for each species, then you say, I'm going to preserve the one that has the highest value, right? So it's the most distinctive, has the fewest close relatives, contains the most unique information. And then I'm going to take the next one, number 2, and I ask, well, how much of the tree have I captured? This would be the percent of the tree that I'm preserving, right? So remember, we're tending to preservation. And it finds out that the Shapley value does, or the ED, so we're using those interchangeably now for the moment, actually captures much more than if you just chose species at random, which is quite interesting. Not as much as if you actually were going out to preserve PD, but there is in fact no measure, there's no mapping of the tree onto the tips yet that is designed to maximize the amount of tree that is retained. Likely because there's no unique way to do that, right? Because you just use a greedy algorithm and move up from the base to the tips, so there's no unique way. The Shapley value actually proportions the entire tree to the leaves. It's not designed to do that, but we can show this correspondence with the way that Dave cut the tree up and gave it to the tips. It is in fact almost exactly a fair proportioning. It captures a lot of PD, which we think is a good thing. And the most important thing is that, semi-independently, the ED measure, this fair proportion measure, is already used in conservation. So I'll show you just the slide for that. So who are the species? Well, you might already think of them. So the most highest Shapley value, most evolutionary distinctive mammal is the platypus. The most distinctive, highest Shapley value bird, we're almost positive, though we don't have the tree completely done yet, is the ostrich. And as you can see, the values are very highly, this is the frequency across all the species, and you can see that they're highly skewed. Now, why are they highly skewed? They're highly skewed because that tree is very unbalanced. So to the extent the tree is unbalanced, your ED measures become more and more skewed. That I think should be clear. Most of the information about the distinctiveness score is actually found in the pen and edge as well, or the first two or three nodes coming from the pen and edge into the interior. Other notable high ED species, the sunbittern. If you get rid of the paleognats, this is likely to be the most distinctive bird, and the tuatara is definitely the most distinctive reptile, as everybody knows already, but you can now quantify that. My favorite is the tailed frog. It lives, so here's Vancouver, this is the west coast of Canada. It lives here, just got split into 2 species. It has a pretty small range. Its closest relatives are all in New Zealand, and they're all highly endangered. And it's number 4 of almost 6,000 species of amphibians worldwide, is the most distinctive species on a very bad tree. Okay, so there is in fact an entire program called the EDGE program, evolutionary distinctive, Shapley value, and globally endangered. So you can actually give money to species that are both distinctive on this measure, a fair proportion measure that Dave came up with, and at high risk of extinction. So for instance, right now we're working on producing a tree or a set of trees for all 10,000 species of bird in order to calculate the ED and the EDGE scores for all the birds so that people in Britain can give lots of money for conservation of birds. Okay. This one is closely related. Instead of taking all equiprobable sets of leaves, we can actually do what we've done before, is we can actually weight those trees by their probability of persisting into the future, right? So as we go through all the trees that a particular focal species actually contributes information to, well, this one's not particularly likely because these guys are both critically endangered, for instance. So this is more likely the tree that it will contribute to into the future, right, to the extent that these are badly managed and go extinct. And so we can actually weight all the trees by their probability of being around, and we get something that Mike Steele was able to compute called the heightened evolutionary distinctiveness. Turns out that the heightened evolutionary distinctiveness is actually just the expected future tree if the species is there, vocal species, minus the expected future tree if the species isn't there. So actually have a very efficient way to calculate this value. So what is this value? What is a heightened evolutionary distinctiveness? Well, we can take a tree. So again, we go back to the tree of lemurs. For all of the lemurs, I just highlighted a few here, but for all of the lemurs, we actually do have the IUCN designation. So whether it's, not at risk, vulnerable, endangered, critically endangered, we can convert those into probabilities of extinction, and then we can feed that in to this heightened evolutionary distinctiveness framework. And what do we find? Well, I'll just give you a couple of examples. So here's Wynne-Edwards sifaka, and this species, it's endangered itself, but more importantly, its close relatives are in fact critically endangered, which means it actually has heightened evolutionary distinctiveness. So if you just look at it under its distinctiveness, it's not particularly distinctive at all, right? It's short pendant edges, it sits in a part of the tree that's lots of redundancy. So it's about 40th of the 67, but it moves right up to 13th if we include the probability of future trees. So, Milne-Edwards, sorry, I was going to say Wynne-Edwards. Milne-Edwards sifaka, then the way that we can think about it is it bears genetic responsibility for more endangered close relatives. And the reason that might be relevant from a conservation biologist's point of view is that here's its range, and here's the range of its two close relatives that are highly endangered. It might be cheaper to manage because there's more of them. So if you take so this is the triage, right? This is the crux of the matter. This is where, it's easy to talk to you guys about this, but we talked about managers about this or the public about this. This is where people become uncomfortable because the idea is that you remove some of your attention from these guys to these guys because the return on your investment may be higher. We all know that managing species that are doing well is much cheaper than managing species that are doing poorly. Just look at the kakapo or the tuatara. Another example that I've already pointed out here is these two guys. So the Avarius arubra is endangered, but its closest relative is actually critically endangered, and they actually sit together on a particularly long branch on the tree. Right? So there's no redundancy there. They're the only two species that contain this information, and this one now has sort of extra genetic responsibility for its close relative, because its close relative is so highly endangered. Okay. What are some issues that we have to think about? One is we don't know how good trees have to be. This is really relevant. One of the, not to you guys, but generally, one of the arguments against taking this approach, most generally, is people say it costs too much to build trees of everything. We got to do conservation now. I can't sit around to produce all the genomes and do all these fancy things to give me the exact edge length and blah blah blah. So, don't talk to me. I'm busy. But it turns out, as I said, because most of the information is, if you look in a tree, most of the information about how much evolutionary information that you have, sorry, I used the word twice, is found near the tips of the tree, right? Because most of this is highly redundant. We know that. Most of the genetic information, evolutionary information, as you go deeper in the tree, is already particularly redundant. So think about it just in terms of how you chop the tree up and apportion it to the tips. These get chopped up amongst everybody. But this is unique to this species, and this is unique to only 2 species. So as long as you get this node and this node and this node right, maybe you only have to go back about 3 nodes in the tree before you get most of the information about whether, in order to calculate, for instance, the Shapley value. Right? So the trees don't necessarily have to be particularly good, but we don't know that. So this summer, I've got a student just taking barcode data. So this very simple, short 650 base pair, building trees, and then comparing the distinctiveness values you get from those very bad trees with actual real trees, or better trees. Another really important question that is very hard to think about even is what constitutes a lineage worthy of attention. This is a debate in Canada right now with endangered species legislation. It's a debate everywhere. What population, what collection do you say this is something that I should be conserving? We talk about, they're called endangered species laws, but they don't conserve species. They don't manage species. They manage evolutionary distinctive units or diagnosable units or subspecies. It's not clear, because as soon as you decide, as soon as someone comes along, and this happens with the lemurs all the time, and says, oh, actually, this isn't, this was well, I don't remember now. You take one species and you say, well, actually, I'm now doing a taxonomic revision. It's now 2 species. I've given it 2 different names. Then, of course, the distinctiveness measure completely changes, because now there's redundancy that wasn't there before, because now you're managing these separately. This is a big problem. Conservation is local, but tree information is global. So you could say, well, we have an endangered species or subspecies in Canada. Well, it's actually not particularly important because it's got a sister species in Mexico that it's very closely related to. But if you're Canadian, you might not care about that species down in Mexico, right? Because conservation is local. I mean, this is a serious issue. Lemurs are a good exception because the entire clade is right there. The survival probabilities that we use for calculating heightened evolutionary distinctiveness or for doing this imperiled PD mapping on landscape may be completely fictitious. And also, very importantly, we have to figure out how we combine evolutionary information of importance with other metrics of importance. Obviously, it's not the only thing that feeds into which bits of biodiversity you should pay attention to. So we have to figure out, this is very hard to come up with a fungible metric that you can apply then in some reasonable way. And that's the biggest argument I get from other academics, is that this is crazy because you're measuring distinctiveness in terms of branch length, maybe millions of years. How do you compare that to some other bit of importance? And to end, finally, and this is something that even came up today at breakfast, is that we think in terms of trees. But I started off with PD, groups of species, and did all sorts of different things, other things on phylogenetic diversity, and then I thought, then I sort of realized that people don't do that. They talk about species. So then, Dave came and we started figuring out, well, how do you map the tree onto species? And so we could rank species. And then I get more involved in the conservation sort of legislation and policy in my home country. And the idea that you would rank species is politically very difficult to do. We don't do that. Either you're in danger or you're not. So you choose your unit and you say, this is a species that is in danger, so we're going to put money into it. At that point, though, managers do get interested in mapping populations on the landscape and trying to decide where they should allocate money on the landscape. For some reason, as soon as because everybody's endangered, so no one can, now you're saying, okay, well, and caribou is the classic example here, is that they spend a lot of time trying to map caribou populations. How are they connected to each other demographically? How are they connected to each other genetically? Well, that is a network, right? So you can calculate, I don't know where the eraser is, but you can calculate, so you can take populations of different sizes of caribou. So pretend this is the northern boreal, and we've got different herds of caribou that have FST values, for instance, between them, or some other measure of sort of genetic proximity. Well, and we've got things like population sizes, so we can actually come up with things like probabilities of extinction on these, or some other metrics. Well, we can actually, should be able to apply the Shapley measure, I just realized a couple of weeks ago, because it's for unrooted trees, on these types of networks. And so you can turn them into split systems and some other things, and that's ongoing research that's been happening in the past couple of days over lunch next door. So that's where we're taking that. And I think ultimately, even though it's the farthest removed from where I started, this ultimately may be the most practical use of all this stuff. Thank you very much. And this way.
Okay, thank you also for staying in time. Questions? Yes.
So I suspect that a negative gamma but unbalanced trees requires that you need interaction between different leaves somehow. And this relates to your HED and so on. The probabilities of extinction might not be uncorrelated. You take that into account?
So we don't take that into account, but that's probably true. Yeah, so we've been saying that the probabilities of extinction come from elsewhere, and we place them on the tips. But it may be that the actual probabilities of extinction are correlated, and the tree might give information about that. Now, the interesting thing is, at least for the lemurs and for the mammals and for the birds that we know about, probability of extinction is not highly heritable on the tree. Much less than I would have expected from first principles, because you think there are life history traits and things that would feed into that, but it's not. But yeah, it's a good point.
So you're focusing on conserving individual species. I know not a lot about conservation biology, but how is it done in practice? Isn't it more that whole habitats are preserved by fencing off or turning into nature reserves than individual species? And
right.
Overlapping and contain themselves, the species would be more...
Right, so there is no legislation in the world that's called an Endangered Habitat Act. Australia has the ability to do something, but almost for some reason, because of the Endangered Species Act in America that was based on tips, everyone else has gone the way of going for tips. So from legislation point of view, no. Practically, yes, people build fences around things.
So if you want to maximize the diversity that is retained, then you should look at the overlap between the different species in the habitats and more select for habitats to preserve and not for individual species.
So right now we're talking about a new endangered species legislation in my home state, and the NGOs are trying to push something where you legislate that kind of thing. It's very hard to monitor ecosystems and monitor habitats. It's easier to monitor species. But yes, that's why I did that mapping exercise. So we do that mapping exercise for lots of different groups of species on the landscape, and then you would find parts of the landscape where you have a lot of information from a lot of species, and then map that onto habitat. Right?
Briefly,
Yep.
sorry, what's the Shapley value for humans?
I don't know. I mean, I did know, but I don't know. I think we ranked like 9th for primates. We're actually quite distinctive because we sit. There's not much around there because those great apes go quite deep.
Worth preserving.
So we're worth preserving. Yeah.
Yeah.
So what choice of branch temp should you use? Because you could use substitutions per site, or you could use millions of years.
Yeah.
And that's quite a difficult question to make a choice between.
That is in fact a very difficult question. So what I've been advocating is that time is fungible across groups. Time and evolutionary information must be correlated, but no one's done, and I've always wanted someone to do this, is to look and see what the actual correlation, but we need to know what the real information content is, but what's the actual correlation between time and information on a tree, and how many genetic markers do you need to capture that?
Mike?
If you included in this analysis the risks that a species has to the survival of others,
Right.
do you think that humans would still be...
No, I mean, they'd have a negative correlation there, and so you'd remove them from the landscape. Yeah.
Okay, is there, well, one short question.
No, just to follow up on that. Do you have species that it's just a biology question.
Yeah.
Do you have species that you deliberately ignore because they're parasitizing other species, and therefore their extinction would actually preserve something else?
So we've only looked at this on sort of birds and mammals, so we haven't run up against that kind of thing. But obviously, a real analysis would want to know the interactions between. But we know very little about the interactions between most species in terms of the demographic effects they have on other species, except for leishmaniasis. I mean, some very particular rinderpest or something.
Okay.
Yep. Thank you.
Arne.